here. Yeah, so you went through the different structures of the nephron in the video. We'll kind of recap that in a second. But let's talk about a couple unique parts. This is kind of more of the functional anatomy of the kidney. One portion of each of the nephrons that's kind of a collection of multiple parts of it is called the juxtaglomerular apparatus. Okay, and that just means like next to the glomerulus, right? Juxtaglomerular. Okay. And this is really the junction of these different spots here. So Bowman's capsule, distal tubule, afferent, efferent arterioles. It's where they kind of come up next to each other. Okay. So sometimes when you see the nephron laid out, this will be kind of flipped and the loop of Henle will be over here, distal tubule over here, and then collecting duct over here. Okay. It's just set up that way in those images just to make it easier to tell what's going on, but that's not typically how it is. It's more like this, where the proximal tubule, loop of Henle, and then distal tubule comes right next to the glomerulus on the other side. Okay. And so we're zooming in on that right here. All right. So this is Bowman's capsule. All right. The glomerulus here, that capillary bed. This is the start of the proximal tubule. You can see those cells with all the microvilli on them. And then we can see where the distal tubule comes up next to the two arterioles here, right? And so the key here is we have two specialized regions that are sensing things for us, and they're helping to regulate the contents of our blood, contents of our urine, and the blood pressure as well. Okay, so there's two special regions. The granular cells are the first one. These have little mechanoreceptors in them, and they are measuring blood pressure. Okay, so we can see those right here. They are around both. We have special cells like that around both the afferent and the efferent arteriole. There's more of them around the afferent, and that tends to be where they really kind of have their primary role is around the afferent arterioles. So this is sensing the pressure of blood going into each glomerulus, right? And we've got like around 3 million of these, right? So this is a lot of different cells sensing the blood pressure going in, okay? And so what they can do is they can release renin. Okay. Who remembers what the stimuli or stimulus for the release of renin was? Low blood flow to the kidneys, right? Here it is, right? Here's the mechanism, right? When blood flow slows down to this area, that means there's less pressure on the mechanoreceptors that are on the surface of these granular cells, and that's the stimulus for them to then release renin into the blood, right? So yeah, so that's one key part here is the blood pressure monitoring system in the kidneys there. It's those little granular cells. And then we have the macula densa cells, and we can see those macula densa cells here, right? So those are in the walls of the distal tubule. Okay, so you see that distal tubule coming up next to the glomerulus here. All right, and so these are chemoreceptors. Okay, and so these are measuring uh, the sodium concentration in the filtrate. Okay, so whereas the macula, or sorry, whereas the granular cells are measuring in the blood, the macula densa cells are measuring in the filtrate, right? So this is like the pre-urine, before we call it urine, it's called filtrate, right? So measuring and sensing the amount of sodium and really the kind of the concentration of the filtrate that's passing through. Okay, and we'll talk about how we're able to then kind of make adjustments based on what the macula densa cells sense here in a second. Okay, but those are the key regions there in the juxtaglomerular apparatus. Okay, questions on that? Okay, all right, and I like this image because it combines gross anatomy with microanatomy. right? And so the gross anatomy part is kind of what you can see here, right? So this would be like the cortex of the kidneys out here, right down into the medulla, the deeper part, right? Um, and we can see where where the filtrate is released into these the a minor calyx like this, and the major calyx down into the renal pelvis, where then collects into the ureter, right? But then we have overlaid nephrons, which are not you couldn't really see, right? Those are not the same size as those things, but you kind of have that overlaid there, so we can see which kind of structures are found in different parts, right? So the cortex is here. All right, medulla is here. Okay, so we can see up in the cortex, um, we're always going to have Bowman's capsule. We'll have the proximal and distal tubules. All right, we'll have the start of the collecting duct or the top of the collecting ducts, CD collecting ducts, and then we'll have those paratubular capillaries. Okay, so those are the capillary beds that are surrounding the proximal and distal tubules. Okay, and then down in here, right, the rest of this is the medulla. So all this part in here, um, we're going to see most of the loop of Henle, most of the collecting duct, and then the vasorecta too. The vasorecta is that capillary bed that goes around the long loops of Henle in those longer nephrons, the juxtaglomerular nephrons. Okay, so it's just kind of like trying to overlay the microanatomy with the macro or gross anatomy there. Okay, questions on any of that or what I wrote? What's your question, Lena? Yeah, what's these ones here? Second one. Most of collecting duct is what I'm saying there. Yeah, thank you for asking. Okay, all right, so here's what I want to do for a sec. I made this into two slides on your thing. It's just one slide, but I just didn't think I would really have room on the next one to draw it. So here's what I want you guys to do, just as a quick recap here. So kind of list the nephron and kidney structures in order from where filtrate is formed to where it's going to end in the bladder. Okay, so this would not be the blood structures, not the vascular structures, but this would be like the tubular structures, right? So starting with Bowman's capsule, that'd be your starting point, like where filtrates form, okay? And then in order, where does everything go? So that again is kind of connecting the microanatomy then to the larger gross anatomy of the kidney, okay? And then here, go ahead and draw out the nephron, um, including the glomerulus and all the tubular structures, and just kind of label those things. And we'll come back together here in a sec, and then we'll dive into more of the functions of those here in a sec. Okay, go ahead. Testing out your art skills this morning. Might need to have one of you guys mirror instead of me to see see a nice drawing. There you go. There we go. I like that. Like that glomerulus. That's nice.
Put this one first. That's probably after.
It's supposed to come all the way and touch.
Wow. Oh, you're right.
Oh, that's, yeah. That's pretty trendy.
It's abstract. So.
Oh, baby.
All right, do a quick analysis of somebody next to you's drawing. See if you can see if everything looks good.
I can't understand that.
It's like, it gets. Yeah.
That's actually not so. I mean, yeah.
How the drawings come out.
Um,
Did you do your drawing? I get to show mine to all you guys, if that's not embarrassing. Mostly face. Beautiful. Yeah. All right. All right, let's go ahead and come back together here. Okay. So here's just kind of the steps in this is really like the steps that the filtrator that the urine takes, right? And so that's all going to start at Bowman's capsule, right? That's where the fluid is pulled out of the blood to create the filtrate, okay? And then it's going to go right to the proximal convoluted tubule, right? Then it goes down the descending loop of Henle, right? Makes the U-turn, comes back up the ascending loop of Henle into the distal convoluted tubule, and then to the collecting duct, right? And then when it leaves the nephron, it's going to then dump into kind of just the open part of the kidneys, right? Which is then the first part would be a minor calyx, which then feeds into a bunch of minor calyces feed into a major calyx, right? And then multiple major calyces feed into the renal pelvis, which is kind of the open part that then connects to the ureter, right? There's gonna be one on each kidney, obviously. Those ureters lead to the bladder, and then one more step. I told you to stop at the bladder, but one more step, then to the urethra, right? Would be the last step with urine formation and release. Okay, questions on that? Got that? Okay. Give you a sec. Yeah. And
Oh, and the glomerulus.
the glomerulus is the capillary bed, and Bowman's capsule is the thing that surrounds it that's like catching the filtrate. And then that feeds into the rest of the tubular part of it. So yeah, good question. Yeah, so glomerulus is a very leaky capillary bed that creates the filtrate, and then Bowman's capsule kind of catches it. Well, good questions. Other questions? Okay, we got that. Let's see a couple people writing. Okay, all right. So here's my beautiful drawing. Not too bad. I gave myself a whole slide. I gave you guys half a slide. But yeah, so yeah, we got the blood coming in, the afferent arteriole, right, into the glomerulus. That filtrate is going to be created, right? So the filtered part of the blood comes out, right, as this liquid that's going to be called filtrate, right? The blood that didn't get filtered or that didn't get pulled out of the blood is going to continue to exit via the efferent arteriole. But now we've got filtrate going into the proximal convoluted tubule, through the loop of Henle, distal tubule, over to the collecting duct. One collecting duct is going to receive multiple sets of filtrate from different nephrons, right? And so we got all these little branches to it there. So yeah, so that's it. Questions on that? Okay. All right. Well, let's talk about a little bit of the function of the kidneys, and we'll start with the innervation side of it. Okay. So there's a reflex in the kidneys known as the renal reflex. Okay. Meaning that's like kidney to kidney, right? That's what that would mean. Okay. And so the way this works is with increased ureter pressure in one kidney. Okay, so this is like increased urine production, right? The ureter would have an increase in pressure if we filled it, right? So we're filling a ureter with urine, right? Sending more urine out of that kidney. Okay, so that stretch is going to be sensed by some stretch receptors, and that's going to create an afferent signal that goes back to the spinal cord. Okay, we send an afferent signal back to the spinal cord, and then the output is going to be decreased efferent nerve activity to the other kidney, not the one that sensed, not the one that was connected to the ureter that had increased flow, but the other one. Okay. So if we decrease efferent nerve activity, so what's the effect of efferent signals? Typically, if we're increasing efferent activity onto a kidney, that's going to constrict the arterioles, okay? And it's always an effect where we get more of that on the afferent side, meaning that we're sending less blood into the glomerulus, okay? So in this case, we're doing the opposite of that, right? We're decreasing efferent nerve activity, so that's going to lead to vasodilation. And again, more so on the afferent side, which then that's going to increase. We'll get to these terms later, but GFR just means glomerular filtration rate. Right? So if we're sending more blood into the glomerulus, then we're going to create more filtrate. That's the bottom line. Okay, making more filtrate. So we're doing that on the opposite kidney that sensed the high flow into the ureter, right? So it makes sense. Like if we're creating a bunch of urine on one side for some reason and not as much on the other side, then reflexively that is going to cause more urine production in the opposite kidney to kind of just balance out the activity of the two sides. Does that make sense? Okay. All right, and then we've got a couple other things going on. So both the proximal and distal tubules and the loop of Henle are very richly innervated throughout the kidneys. Okay, and so stimulation of these areas can happen via norepinephrine on adrenergic receptors. Okay, both on the tubule cells, those are just the ones like lining the tubes. Okay, those are the ones that are going to be doing the movement of water and sodium and all of that we'll be talking about. But then also those granular cells that we were just talking about. Okay, those granular cells are the ones sensing the blood pressure, right, in the afferent arteriole, okay, and efferent. And so what are some effects of this is really like sympathetic nervous system activity, right? We're activating the sympathetic nervous system. So what are some effects of that? Number one, we would increase the sensitivity of those granular cells. Okay, so making them more likely to respond and release renin if there's a decrease in flow. Yeah, Tip?
That's afferent or efferent with the E after the dot is highlighted.
Oh, that says afferent. Yeah, thank you. Yeah, good question. Yeah. Okay, so we're increasing sensitivity to the granular cells. We're increasing reabsorption of sodium. Okay, and then we're getting renal vasoconstriction as well, which would then decrease our GFR, meaning we make less filtrate and decrease renal blood flow as well. Okay, so if we think about this, all of these things are slowing down urine production. Right? So we're slowing down urine production. If we're increasing the sensitivity of the granular cells, that means we're going to release renin more. We're more likely to release renin. The results of releasing renin are what? What happens when we release renin? Maybe check in with each other on that real quick. Remind yourself of that chain of events. Okay, so which hormone gets produced when we release renin? What's the kind of end result as far as the hormone goes? Angiotensin 2. Yeah, angiotensin 2 has powerful effects on what? Blood pressure. Yeah. And so which way, up or down? Up, yeah, for sure. So we're holding on to more fluid, we're holding on to more sodium, we're constricting vessels all throughout the body. Okay. So increase in sensitivity of those granular cells is going to increase our blood pressure by doing those things, right? By holding on to more fluid, holding on to more salt. Okay. We're also going to directly hold on to more sodium or at least pull it back from the filtrate, right? That's what this is. And then we're going to create less filtrate from the get-go, right? By renal vasoconstriction. And so all of these things make sense with what we learned before about the idea that the sympathetic nervous system causes a decrease in production of urine, right? That's what's happening here, right? These are just kind of three mechanisms as to how activating adrenergic receptors throughout the kidneys will decrease urine production. Okay, yeah, maybe. Mm-hmm. You talking about in the afferent and efferent arterial? Yeah. We'll kind of dive into it on the next slide a little bit too. Yeah, yeah. But ask it again if you didn't feel like I did it justice. Okay. Good question. Other questions on this right here? Okay. All right. So then let's talk about the filtration process. Okay. So filtration, this is what our kidneys do, right? This is what our kidneys are known for. This is their primary function is to filter our blood. Okay. So we get about 1.2 liters per minute of blood flow going to our kidneys. Okay. That's a lot, right? When you look at our total cardiac output at rest, about 5 to 6 liters, right? This is a fifth to a quarter of our blood flow going to our kidneys, right? So it's a lot of fluid. Okay. So this is 1,500 liters of blood going to our kidneys per day, which is 400 gallons, right? This is a lot of fluid going through our kidneys. Okay. So that's just blood flow to it. But then of that blood flow, about 120 to 125 mils per minute is actually pulled out of the blood, right? So that's the filtered part, okay, which amounts to 180 liters per day of fluid that's pulled out of our blood, which amounts to 47 gallons of blood that's pulled out of our, or fluid that's pulled out of our blood per day, okay? But we only urinate out about 1.8 liters, okay? So what percent of the total filtrate is that? One, that's it, right? So 1% uh, of the total filtrate is actually released from the body. Okay. And that's how we want it, right? We're doing this filtration thing so we can clean the blood, we can pull things out of it we don't want, we can put things into the urine that we want to get rid of. But we don't want to lose all that fluid, right? That's not the point of this, to lose a ton of fluid. There's certain things we need to get rid of, like nitrogenous waste products, right? And some hydrogen ions and things like that. But we don't want to lose all that fluid. If we lost, obviously that wouldn't work, right? We don't have that much fluid in our body, right? We don't have 180 liters of fluid in our body. So there's no way this would work, right? So we got to pull most of it back. And so that's mostly what we're doing in the kidneys, is pulling most of it back, right? But we need to create a bunch of filtrate first, okay? So the question here is, how do we create so much filtrate, right? And so there's some anatomical contributions to this, and this idea of structure dictating function, right? Which is kind of a key through all of anatomy and physiology. And so one of those anatomical contributions are the renal arteries, right? We got these big renal arteries. You guys have probably seen those in the donors, right? How big? How big is it? Size of your finger, bigger? Show me with your hand. Pinky, yeah, yeah, yeah. So not huge, but yeah, maybe something like that. Okay, but pretty big and pretty short too, right? They come right off the aorta. So we got tons of blood pressure in the aorta, right? And then this pretty sizable artery coming right off, and it's short, right? One of the main contributors to resistance of blood flow is the length of a blood vessel, right? And so we got short, large diameter renal arteries, which is going to increase flow, right? Really, that means increase hydrostatic pressure, right? Because we're kind of leading up to how do we get so much fluid to leave the blood in that capillary bed called the glomerulus, right? And so the two factors we talked about before with fluid movement in a capillary bed are hydrostatic pressure and osmotic pressure, right? So we need to have a lot of hydrostatic pressure. And so that really helps with that, right? A big artery that comes right off the aorta and it's short, okay? Secondly, there's this idea of these two arterioles. So one that's going into the glomerulus. Okay, so the glomerulus is this just wound up kind of mess of capillaries, right? It's very curvy, and they're very fenestrated, which we'll talk about in a second, meaning there's lots of holes between the cells, between those endothelial cells. Okay, but the one interesting dynamic here is that the afferent side is bigger than the efferent side. Okay, so it means we're sending in a lot of blood to the capillary bed, and then it kind of gets backed up in there, right? Because it's like trying to leave the other side, but it its ability, the vessel that allows it to leave is smaller than the vessel that lets it come in, right? So the blood kind of gets backed up in there, all right? So this also plays a role of increasing hydrostatic pressure as well. Okay, and then the capillaries are very leaky, right? So increased permeability. All right, so fluid can move through. Okay, so then kind of to your question, Ava, I think if we're talking about this afferent and efferent arterial, when we send sympathetic nervous system activity to the kidneys, we constrict both sides, but there's a lot stronger effect on the afferent side than there's the efferent side. The efferent side's already smaller, right? So we've already got the smaller tube and bigger tube, right? And then we constrict them both, but there's more of an effect on the afferent side. And so we're always going to be, it's easier just to think about it in terms of the afferent only, probably, that if we constrict, then we're sending less blood into the glomerulus, which would decrease hydrostatic pressure, which then we would decrease glomerular filtration rate. If we take some of that off, right, if we remove sympathetic activity, then we're dilating. So we're sending more flow into it, creating more hydrostatic pressure, creating more filtration. So even though the other one is affected, it's affected less. So it's kind of just easier to think about the first one. Yeah. Does that help? Yeah. Okay. Questions on that part at all? Okay. I think the other anatomical factor to think about here is that we have around 3 million of these, right? So that helps, right? There's a lot of them, a lot of this happening all at once, right? And so then what does that filtrate look like that we actually make, right? What is the contents of the stuff that gets pulled out of the blood? Well, there's no blood cells typically, and there's no proteins for the most part because those things are too big to get through those fenestrations in the capillary walls. Okay. There are in some cases, right? If we have some damage to the kidneys, if we have some other things going on, then we can get some blood in our urine, we can get some proteins in our urine. But under normal conditions, we don't really have that. Okay. So it's mostly water, small molecules, very similar to our plasma minus the plasma proteins. Okay. All right, so then moving on to this idea of glomerular filtration rate. Okay, so this is just like the rate of filtrate formation by our kidneys. Okay, this is known as GFR, right? Super common measurement that you'll see on all your patients, right? It can be measured multiple ways. We'll talk about some of those next class, but a normal GFR is about 125 mils per minute. Okay, that's the same as what we were just saying on the previous slide, right? 100 to 125. That's the same number there. Okay. Um, Once it gets below about 60, then we start having kidney problems. Okay, which is pretty amazing in a way, right? Where we can lose over half the function of our kidneys before we really notice any problem with that, right? Any symptoms. Okay, so overall, this GFR stays super, super constant, and the ways that it stays constant are a couple of them here. So we have auto regulation of blood flow to our kidneys. Okay, so if we all of a sudden have this big increase in systemic blood pressure, this would increase our GFR, right? If we're just all of a sudden increasing the speed of blood flow around the body, then we're going to create more hydrostatic pressure. We're going to create more filtrate, right? This is just what happens. So we'd increase GFR, okay? And so the kind of immediate reaction to this is the afferent arteriole constricts. Sorry, I ran out of room there. I just wrote constricts. Okay, so afferent arteriole constricts. And then that's going to drop our GFR back to normal. This is just kind of the same thing that we talked about in our all smooth muscle. That's just kind of the way it reacts when we send high pressure to it. It kind of stretches it out, and the reactiveness of the smooth muscle just clamps down on that. Right, it's the same idea. So this is myogenic response. Okay, and it's just the opposite if we have a decrease in systemic blood pressure. Right, this decreases our GFR. This is going to lead to dilation instead, right? And this is going to bring our GFR back up in response to that. Okay, so this is part of the regulation. It's just kind of the properties of the smooth muscle cells and how they respond to pressure changes. Okay. We also regulate our GFR via changing the permeability of those capillaries in our glomeruli. Okay. And so substances that don't have a charge, kind of between this size here, 4 to 8 nanometers, can freely pass through. But our capillary walls have lots of negatively charged proteins in them, so that's going to block things like albumin, which would fit through that size, but that negative charge kind of repels them. Okay, positively charged substances can get through easier, but then capillary permeability is changed by the contraction of those mesangial cells. Those mesangial cells are very similar to the parasites we saw in a regular capillary bed, where they just kind of are these little contractile cells that wrap around the capillaries, and when they contract, they make the holes, the fenestration, smaller. They're just pulling the cells together, right? And so one of the mechanisms for contraction of these cells is angiotensin 2. Angiotensin 2 leads to contraction of these cells, which then that's going to decrease our GFR, right? And again, angiotensin 2 helps us to hold on to more fluid and raise our blood pressure, right? So this is one way that angiotensin 2 does that. Okay, questions on that? Yeah, go ahead, Bill.
The dilation only happened in the afferent arteriole, not the efferent.
Um,
Is that just the regulation?
In this case, yes, because of the way that we're just kind of, if we're just like sending more blood flow into that area, it's going to stretch out that afferent arteriole, and then it kind of we lose most of our blood pressure through the glomerulus anyways, right? And so we don't have as strong of an effect on the other side because the blood pressure is mostly kind of lost at that point. Yeah, good question. Okay, other questions? Okay, so let's look at one more mechanism for changing the concentration of filtrate. And so this is known as tubuloglomerular feedback. Okay, and so tubuloglomerular feedback is kind of kicked off when we have increased blood flow to our glomerulus. Okay, this is going to increase GFR, right? So we can kind of go with the steps here. Which one is cut off, but you can see it right here. That should have been the one there. Okay, so we can see one there. So we got increased blood flow into the glomerulus. Okay, that leads to increased GFR, right? Because we're just increasing hydrostatic pressure. Okay, When we have extra filtrate being formed, that means we also have extra sodium in our filtrate, right? Because we're pushing more filtrate into the tubular system, okay? And that's going to be sensed by those macula densa cells, okay? The ones we were just talking about, which are the kind of the chemoreceptors of the kidneys, right? This thing is kind of outlining the mechanism of how those macula densa cells help us control the concentration of the filtrate, okay? So that leads to diffusion of sodium chloride into those macula densa cells, okay? Water's going to follow those. Water always follows the sodium, okay? So we see increased sodium chloride delivery into the cells here, right? And then that water follows it, so then it makes those cells swell, and that mechanical swelling of the cells opens up ion channels that allows ATP to leave those cells. This is a kind of an interesting different mechanism than what we're used to seeing, right? So ATP is leaving the cells, okay? Outside of the cells, it gets broken down, which results in the formation of some broken down ATP, which is adenosine, right? We have adenosine receptors all over our body. Adenosine receptors are super common, and this is an example of one, okay? So now we have increased levels of adenosine, okay? So we're activating adenosine receptors on those mesangial cells again, and smooth muscle, and the response to those two types of cells when adenosine binds to them is they cause intracellular calcium release, and that leads to constriction of both the afferent arterioles, okay, and contraction of those mesangial cells, which leads to a decrease in glomerular permeability. Okay, so the end result of that is this right here. So vasoconstriction and contraction of the mesangial cells. Okay, so we'll put a number 7 there for that one. Okay, so here's what I want to do. That you'll have to kind of take some time going through that, and I want you to do that here in a sec. But let's go ahead and just recap these kind of important cells that were from the video, because we'll continue to talk about these. Now I also want you to outline the flow of blood through the kidneys. We outlined the flow of filtrate in each structure first, but what about flow of blood? Okay, you can kind of recap these anatomical features we just talked about, and then I do want you to kind of run through those steps we just did, talk that out with your partner to make sure you're wrapping your head around what's going on there. Okay, so let's just come back in about 11 minutes after doing this and taking a short break. Okay, and we'll start back up at 9:30. Okay, let's go ahead.
I'll be freaking out.
All right, guys, we're going to go ahead and get started again here. I thought I had the screen frozen, but I didn't. So you guys have all seen what I wrote, but that's fine. So any questions about the top part here, or any questions about what I wrote there as well? Don't worry about all the little arteries and venules in that image of the kidneys to memorize those. You don't need to know more than what I have here. The one thing that's a little bit tricky as far as like the order of things, and again, don't worry about it too much, is just like the paratubular capillaries versus the vasorecta. We really only have the vasorecta on those juxtaglomerular nephrons, which are the ones that have the deeper and longer loops of Henle. All of them have the paratubular capillaries. So that's why it's kind of like a and or situation with those capillary beds. So questions about any of that stuff? And then with the tubuloglomerular feedback, that was just kind of obviously what we just went through here. But any clarifications needed on this? Is there any part you want me to go through again, or you have questions about this? Yeah, Joseph.
So ATP just comes out from.
Yeah, so probably a concentration gradient situation, where there's going to be more inside the cell versus outside. Yeah. And ATP is a pretty big molecule, so they must be pretty large channels to allow that to go through. Yeah. Other questions on that? Yeah.
Should we
Yeah.
also like pay attention to where this is all happening? Just like associating the locations and each thing.
Yeah, so this is mostly this is all happening in that juxtaglomerular apparatus for the most part, right? Because that's where the macula densa cells are, right? And that's not where the filtrate is formed, right? That happens first, but we're sensing this is the chemoreceptor part of those macula densa cells, right? This is like kind of outlining the mechanism of how they're chemoreceptors, right? How they're sensing the amount of sodium in the concentration of the filtrate, the amount of sodium in the filtrate is happening because of this mechanism here. So yeah, this is for the most part happening in the distal tubule, right? And then the cells, and then those macula densa cells lining the distal tubule is where that's happening. Yeah. Question, Andy? Yeah.
Happening exclusively in the distal tubule after the reabsorption of the stuff has already happened.
Yeah, totally. So, so we're talking about right here, right? This part right here, back on that first slide. And the reason it's important right here is because there's just so much of it. So like tons of concentration of sodium and water in the first part, right? So the distal tubule is kind of always, we think about that as like the fine tuning of the filtrate, right? Like, do we need to pull a little bit more sodium out? Do we need to put a little bit more back in? And so this is kind of our opportunity to sense the filtrate and make that fine tuning take place, is right before it goes to the collecting duct. Good questions. Other questions on that? Yeah, Andy, go
Is
ahead.
there an area where the water is primarily absorbed? Or is it kind of
It's
just depending on?
Well, it's everywhere sodium is for the most part. We'll get into the details in a sec. The vast majority of it happens in the proximal tubule. That's the vast majority. It's like 60% of all the water and sodium comes back in the proximal tubule. We get some in the descending loop of Henle, not in the ascending. We get just a little bit in the distal tubule, not much. And then finally, a good chunk in the collecting duct tube. We'll go through all those details, probably most of that, a little bit today and then most of that tomorrow. But yeah. Yeah, good. Other questions? Okay. All right. Well, let's go ahead and move on to starting to talk about each part of the nephron and what happens. Okay. But before we do that, kind of just need to make sure we have our heads wrapped around these terms that we'll use. Okay. And so we've already talked about glomerular filtration, right? This is just formation of filtrate, right? This is pulling that fluid and all the stuff in the fluid out of the blood. Okay, so that's what filtration means, glomerular filtration. Okay, and then the other important term is tubular reabsorption. Okay, so this is going to be bringing things back. Bringing substances back from the filtrate. And they're eventually going to get back into the blood. Okay, so think about everything that went into the filtrate in the glomerulus or through the glomerulus into the Bowman's capsule. There's a lot of stuff that we want to get back, right? We don't want it to be lost into the urine, right? So all of that stuff we would consider reabsorbed, right? So it had to be put into a space in the body that's continuous with the outside first, and then when we get it back, it's reabsorbed. Okay. And so that filtrate is starting isoosmotic with the blood, so there's no concentration gradient. So anytime we want to get things back, initially we're going to have to use active transport because we don't have a gradient, right? Because it's the same gradient of same amount of sodium in the filtrate that there is in the water relatively to the volume of water, right? The same amount of filtrate, same amount of sodium that's in the filtrate versus the blood. I think I said the water, relative to the amount of water that we have. Okay. And so all of this is driven by membrane proteins on the little cells that line our kidney tubules, right? And all of these have a maximum rate that they can work with, right? And so a great example of this is in type 2 diabetes, really also type 1 diabetes as well. But we have certain glucose transport channels that are working hard to pull all the glucose out of our filtrate. And most of the time we don't have glucose in our urine, right? But in type 2 diabetes, type 1 diabetes, we can, right? Because we max out the rate of those pumps. And so glucose is going to pass without getting grabbed and reabsorbed as it passes through. Okay. And then that creates osmotic pressure in the filtrate, right? Which then pulls a lot more water with it too, right? Which is why we increase the volume of our urine as well, is because we're not able to get those osmotic agents out as properly. And so water's going to stay with them. Okay, so that's reabsorption. And then secretion is the opposite. These are things that we're adding to the filtrate. After initial formation. All right, so we have the initial formation of our filtrate up here, and then there's certain things that wasn't enough, right? We want to get rid of more of that thing than came in originally, so we secrete it into the filtrate, right? So tubular secretion is the term for that. Okay, these are mostly things that the body wants to get rid of, right? Okay, and then all of this together will have an overall effect on two things that we'll talk about as we go through each part of the nephron, mostly next class, which will be the concentration of the filtrate or concentration of the filtrate and urine, right? But then also the volume of the urine. Okay, so it's like how much did we start with? How much did we bring back? How much did we add as we kept going through the kidneys? Okay, questions on any of that? Okay, so let's talk just briefly about sodium reabsorption because it's such a key aspect of everything that the kidneys do. And so, first of all, sodium is pulled into the kidney tubule cells by a bunch of different mechanisms. Okay, a lot of these are co-transport mechanisms. Okay, so lots of different types of proteins that line the walls of the kidney tubules. Okay, some of these are co-transport with glucose, some with phosphate, amino acids, lactate, hydrogen, chloride. All of these things are different, just different mechanisms that we do this. Okay, and so that's all being represented by this, right? Pulling it out of the lumen of the tubular part of the kidneys into the cell. Okay, and so there's no like mechanism outlined there because there's just so many different ones. Okay, but then secondly, we've got to get it from the inside of that cell over to the interstitial fluid so then it can get into the blood and get back to the body, right? Or back to the bloodstream. Okay. And that's always done via the sodium potassium pump at that point. Okay. So we get it into the cell by a whole host of mechanisms, but then we get it out of the cell over that interstitial space where then we're going to have a capillary over here, right? Where that sodium is going to make its way to. We get that always through the sodium potassium pump. Okay. And as we were just talking about, 60% of sodium reabsorption happens in the proximal tubule. This is where most of it happens. We get about another 30% in the ascending limb of the loop of Henle. This is the thick part of the loop of Henle. We get a little bit more in the distal tubule and then a little bit more in the collecting duct to get our kind of total amount of sodium that we reabsorb. Okay. And questions on that? Okay, so one thing to note, sodium never like passively gets through the walls of these cells or between the cells. It's always actively transported via some mechanism or another. Okay, so let's start out with what's happening in the proximal tubule. Okay, we'll go through each part of the nephron. So here's our Bowman's capsule, right? We're focusing in on the proximal tubule here first, okay? So again, the filtrate is going to start isoosmotic with the plasma in Bowman's capsule, okay? And then the questions for each little section of the nephron is what's reabsorbed, what's secreted, and what's our overall effect on concentration and volume of the urine, okay? So what's reabsorbed here? Lots of sodium, right, via active transport, and then the water is going to go with it. So it's really the same percentage of water that we bring back. And so, yeah, it's 65% of of the water that we're going to bring back comes back right here in the very first step. Okay, because it follows the sodium. So water doesn't need special channels. There are channels that helps it get through, but yeah, it's the aquaporin channels. It doesn't. What I meant to say, water doesn't need active transport. It does have channels, but it doesn't have special channels that uses ATP. It uses aquaporin channels to get through. Okay, and then we bring back all of our nutrients here too. So any nutrients that ended up in the filtrate, glucose, amino acids, all of these things are brought back in this very first step. The body doesn't want to lose these things, so it's going to pull it all back. Okay, so this 65% equates to 117 liters per day of water that's brought back right here in the proximal tubule. Okay, so it's an equivalent percentage of ions, so that means there is no effect on osmotic pressure. Okay, so that's the reabsorption piece here. The secretion part, we can get rid of some hydrogen ions here if we need to. All right, so when pH is low is when we'd want to do this, right? We'd want to secrete some hydrogen ions, help us to balance pH. Okay, so our overall effect on concentration and volume. So we have a major volume reduction. Right, it's at 65%, but then no concentration change. Okay, so no change in osmotic pressure of the filtrate because kind of an equivalent amount of sodium and water is coming back, but major volume reduction here. Okay. Questions on that? Yeah, Joseph?
Concentration of what, the urine?
Yeah, so all these slides are going to be set up this way. It was what is this part of the kidney tubule cell? The kidney tubules reabsorb, what's secreted here, and then what's our overall effect on the concentration and volume of the urine um, based just on the changes that happen here in the proximal tubule. Yeah, thanks for helping me clarify that. Okay, other questions? Okay, so let's get into the loop of Henle a little bit now. Okay, so we've got the loop of Henle is both the descending part and the ascending part. Okay, so one of the mechanisms here, which we'll talk about on the next slide, is called the counter current multiplier mechanism. But this uses fluid flow in opposite directions because we have fluid going down here, okay, and up here. And these are little microscopic tubes, right? So they're very small and they're right next to each other. And that, along with we'll see on the next slide, but we see the vasorecta is also there too. And we have fluid flowing in opposite directions there as well. What ends up happening is we kind of create this little whirlpool effect in our nephrons, okay? And we'll get to that in a second, but let's talk through this first, okay? So, so the descending limb, it's asking here what's going on as far as concentrating the filtrate. Okay, so in the descending limb, the filtrate is becoming increasingly hyperosmotic versus the blood. Okay, and that's because the descending limb is highly permeable to water. All right, so as the filtrate's going down this way, water's leaving, right? So that means it's getting more and more concentrated with solute, right? Because water is leaving, so we have a higher percentage of solute versus water as it's going down, okay? Then it's going to go down and make that U-turn and start coming back up. And now it is going the other way. So only sodium is leaving. Sorry, we can kind of fill this one in too. This one doesn't transport sodium. In the ascending limb, we get active transport of sodium in the interstitial space, but it's impermeable to water. All right, so this one is moving in the direction first of increasingly hypoosmotic. Right, but first it's kind of like getting back to isoosmotic, and then it's going to even go a little bit past that. So we're losing water here, right? And then we're pumping sodium out over here, so the osmotic pressure is kind of catching up to where it started again. But it's even going to go a little bit past that, and we're going to dilute it a little bit by the top. Okay, so the end result of this is the interstitial fluid around the loop of Henle becomes very hyperosmotic. So this space down here becomes very hyperosmotic because we're adding a bunch of sodium to it. And we'll kind of talk about this on the next slide as well. Okay, so what do we do in terms of volume and concentration changes here? We have another 27 liters per day, which amounts to 15% of our water is reabsorbed here. And as far as the concentration goes, we have a slight dilution of the filtrate. Okay, so the filtrate got very hyperosmotic when it got to the very bottom. And then as soon as we start getting to this thicker part, we start pumping sodium out. So it starts getting less and less osmotic pressure to the point at which it gets back to isoosmotic with the blood, but then it keeps going and we get rid of more sodium. So it actually dilutes the filtrate a little bit there. So we're adding more sodium to the space around the loop of Henle than we are adding water to it. And so the end result of that is this space around here becomes very hyperosmotic and very highly concentrated. Okay, aquaporin 1 is the type of channel that is responsible for most of the water movement here on the thin part of the loop of Henle. Okay, questions on that so far? Okay. I think instead of trying to squeeze in the countercurrent multiplier mechanism in the last few minutes, let's go ahead and just pick that up tomorrow. Yeah, so I think this is a good kind of starting point, intro to what we'll talk about next time. We'll get into this stuff tomorrow and we'll finish the rest of this up. So let's stop there. Have a great rest of your day. Good luck on your exam today, guys, and we'll see you tomorrow morning. Great.